Hello out there. Welcome to the Gen Bio Lab Practical Review. So we're gonna stay focused on Gen Bio today, and then it'll be about an hour that we'll be here reviewing, and we'll try to answer like what your questions are. So feel free to comment what your questions are, or say hi that you're here. What I was hoping to get started with is brainstorming some of the topics that you all think are going to be on the practical, so then we can see what you're expecting to be on the practical, what I know is potentially on the practical, and then use that list to decide where we want to start reviewing. So if you're out there watching, feel free to comment some of the things that you're thinking are going to be on the practical. So I'm going to be getting a document ready and we'll compile this list together. So some of the things are going to be pretty similar to what you did see on your mini practical. So let's see, that first question on the mini practical that we just had a few weeks ago was about gram staining. And then we also had some kind of like follow-up questions, right, about like bacterial resistance or antibiotic effectiveness, right? And we also looked at, that also was about the antibiotics modes of action. So those are all things that you will want to make sure that you review. So don't hesitate to throw a comment out of ideas. Let's see, what else was on the mini practical that we had? So there was some genetics practice, right? So that was like looking at the gel. And that gel was a gel electrophoresis, right? And so determining inheritance or like a lot of times we look at a gel for like who is the father of an individual, right? Let's see, what else would have been on the mini practical that you guys had. Of course, the whole back page was writing a results section. So our writing thing, so that might also lead to writing a conclusion statement. And of course, also with that is figure labeling, like in terms of putting those letters on, right, to make sure that we have the proper letters to show our significant differences there. And also a proper figure caption to go with that. And other topics were about like the cell cycle, and so how those cells might look under a microscope if we were able to stain them. Hopefully you guys can chat in and provide some answers and or ask some questions. And then, so this was both the mitosis cell cycle, but also the process of meiosis, right? So just generating new cells versus creating gametes, those sex cells, which kind of leads into our genetics lab that we had earlier this year. And then, of course, we have our math. So we have our lab math, which this semester was a dilution factor, cell concentration, and then just like determining dilutions overall, which we use that C1V1 equals C2V2 equation for that. And when we were looking at that dilution factor and cell concentration, that was the week that we used a laboratory tool called a hemocytometer. So you'll need to know how to use that for your lab practical. And then, the week that we looked at these dilutions was when we were looking at Logger Pro, a program on our computer, and providing, this was our enzyme week, right? And being able to measure with that an initial rate of reaction. So if anyone is out there and listening and wants to comment in, what of these do you want to kind of review first, if anything? Otherwise, I will just go in an order I have pre-planned. Okay. Looks like no one here has any ways that we definitely want to go. I'm appearing to have some technical difficulties. One second. Okay. So first thing that I had prepared, sorry for the technical delays there, was the hemocytometer under the microscope, because many only got limited practice with this for the 1 week in lab. And so just a reminder that Sundays from 5:30 to 8:30 up here in our Gen Bio labs, so that's Johnson 244, there's always TAs available, and you could come in this Sunday or the following Sunday before your lab practical to practice trying to adjust the light to be able to view a hemocytometer grid under the microscope. Often there's also a maximum magnification that we normally go up to, and that is 100 times total magnification. So on our objective lenses, that are the part that we rotate often to change, remember that our ocular lenses, the part that we look through, has a 10x magnification already. And so your 10x on your objective lenses, the ones that you rotate, was probably the best one to go up to. And that lets you see the entire major square as we talk about it under a hemocytometer, right? And so there are 3 ways that you can adjust your light to view the grid. Does anyone know of those? You can comment them in. So the first one should be really easy, right? It is just the on-off switch, right there on our microscopes. I often recommend that the best like light level in terms of like, remember these lights on the side will the lights that are all the way on the far side of that microscope, they will light up depending on how much light you're giving it. And so I recommend when you're trying to find that hemocytometer grid under the microscope that you don't have that light any higher than 3 of those blue dots lit. And that's just because the grid on our hemocytometer slide is very faintly etched onto that slide, right? We can't see it with our naked eye on that slide, but we know it's there when we look under the microscope. And so having too much light creates, like, you know, when you're like trying to look into the sun and you can't see the stoplight out at an intersection, it's like the light that the microscope is catching through the lenses is too much that you don't end up seeing your grid. And so one of the other things to change is what's known as the iris diaphragm. And so it's this little basically like handle that's like under the microscope. That white line or white arrow is trying to point to it. So hopefully you can see that. And that is gonna give you the most minor adjustments. So I always like to start with that all the way closed, which would be all the way to your right hand side. And then as I'm looking through the microscope, just give that like slight taps to just tick it towards open bit by bit until I'm seeing that grid. If I've pushed that iris diaphragm probably halfway to 3/4, what's occurring is likely that my condenser at the bottom was closed too much. And so that is just, it's kind of like opening a jar, but it just has like a part on the side that will move and it will create like a shutter open and close to have more of that light from the light bulb actually coming up through the microscope that you're seeing. So I would say on-off switch, condenser is really what you should change second, and then the iris diaphragm would be what you change last to make those most minor adjustments. And you, of course, should always start looking for your grid on 4X, and you'll probably see the entire grid, right? And so that entire grid looks something like this. And so our major squares, let me grab a contrasting color here, right, is 1, 2, 3, and those are the 4 corners that we're looking at. And if you, sorry, my camera was smaller. In these other areas that I haven't circled, the lines are much more close together, creating more than 16 squares that you would read off of. And then what's nice about our hemocytometers is you'll notice that something like these lines will continue going even out of the center portion of the grid, but there will be, to like close off this square, a little close grouping of 3 lines, and the same for the other side that's like outside of it. And so that will really help you know like where you are on the hemocytometer and making sure that, you know, if you look for those 3 groups of lines, you'll know that they're always on the outside 2 sides. So then you know which of those major squares that you are in. So the kind of connected portion to this, right? We use this hemocytometer to count the cells that we're looking for in some solution, right? And so always feel free to be commenting in any questions you're having, or if I need to slow down or repeat something. And so we have to think about, okay, well, these cells aren't going to magically be directly in these grids that we've created on our slide, right? We're just putting some liquid on the square or on the slide. And so we need to make sure that we know which cells we should count and which cells we shouldn't count based on where they are. So on this slide, and I will actually make it full screen, turn my camera off for a second. In those pink boxes there, hopefully you can see, is the edge of a line that is creating the grid. And so we have to think about, within those pink boxes that I've created, which of those cells would we count and which of those cells would we not count if we're just trying to count any of our cells. So if you have an idea and you want to describe one that would or would not count, or how you're going to determine if something would or would not count, I'll give you a second there. And so the cells, remember, are these kind of like shiny spots that are glowing back at us, not the darker splotches on the slide. And I can tell based on how some of the other lines keep continuing. I believe in this hemocytometer, I am on the lower right-hand side. And I would say that's a pretty good density of cells. Like, there's not going to be too many to count. I know some of the squares of that major square are cut off right now, but generally, right, we look for something to be less than 150 per major square to be considered countable and reasonable to count with our eyes. And so I would say that on the left-hand side, since you guys can't see my mouse, that bottom cell that's in the pink square would not count, as it is a majority outside that boundary line for our grid that we're looking at, the square that we're looking at. And then the next one up would count because it's a majority inside the square that we're looking at. And then that top one all the way to the left, I would say would also count because it is right on the line that we're looking at. And so it should probably count. Others may not count it, and that's how we get off by maybe a cell, and that is okay in the grand scheme of things. And then, of course, the top cell would also count because it is a majority inside the grid that we're looking for. So in terms of counting things, right, we get a count, but a lot of times we have to dilute our solutions enough to make a cell culture countable on our hemocytometer. And so to ensure our correct cell concentration, we use the cell concentration equation. Does anyone recall what some of those parts are? I did have a hint on the slide that it includes our dilution factor, which requires sometimes a separate equation to calculate. Or even just what is the unit that we need to get back to for cell concentration. So we know it's got something to do with our count, right? And so we've talked about, right, we've counted cells across all of these different major squares, but we might find that there's 15 up here and 16 here and 17 here and another 15 here, right? And so what we will do is we will take an average number of cells in the four major squares, MGJR for major, SQ for square. And then, so that gets us our number of cells, right? We're looking for a cell concentration. Concentrations are units that are something per something. So we know that our unit here is going to be cells per something. And so does anyone know what that per is? What are we looking for? Are we looking for cells per liter, cells per milliliter, a cell percentage? What are we looking for there? I'm just adding this DF to stand for dilution factor, which is something that I said, and we're going to multiply them. So the answer is we're looking at cells per milliliter, ml for milliliter. So the way that we will we need to get back to something per milliliter. So we use dilution factor as part of that. I know this is a little small right now, but just hold on for, bear with me for a second. But we also know that there's a specific volume that fits under the cover slip that we put on top of our hemocytometer for the specific major corner area that we are getting an average count of. Now, it's possible if you have smaller cells or something like that, that you count some of these internal areas that we don't count, and those will have a different area that would be there depending on like what part of those parts that you count, right? So we happen to know that this volume that fits under the cover slip is 10 to the, yeah, it's 0.0001 milliliters, which is 10 to the negative fourth milliliters. So one second. So to get our cell concentration equation back to cells per milliliter, we've diluted it to make it 10 to the negative fourth. So to go backwards, we need to multiply by 10 to the fourth milliliters over milliliters. Sorry. And that will create our number of cells per milliliter. So we also talked about this dilution factor. And what is that? So we say that the dilution factor is equal to V2 over V1. Don't forget, you guys can always comment in some questions about things so I can make sure that I'm explaining them, right? This review is for you. And so what do these V's mean, right? We could probably assume that if there's a V, it probably either means volume or velocity. And in bio, we're not working with velocity. So we know these V's are volumes. But what are they volumes of? So V2 is the volume total in basically in the tube, right? Because we use various size tubes, which have specific names, but we never talk about those specific names in lab really, where we're taking some of our cell culture and we're taking some sort of buffer, whether that be PBS buffer like we did in our yeast lab. Some of you who did dilutions for your for your independent projects might have used just distilled water or another type of buffer or dechlorinated water. So we just say buffer as a general, right? It's basically what we dilute with. So the total volume in the tube is what's going to go on top. And this V1 is going to be the volume of transfer from last tube with cells. Basically, the volume that you are going to take from one tube out with your micropipette and put into your new tube. And that we're assuming, like, to begin with, of initial dilution, this would be a very highly concentrated cell culture, right? And we know we need to dilute it to be able to count it under this hemocytometer so that it's easily countable with our naked eye. But sometimes we have to do multiple dilutions in order to get to a point where something is countable. For example, I know that some of my students' independent projects who are working with algae, I said, okay, let's do a 10 times dilution, and they still were seeing too many cells, so we cut it in half, right? And so now they did from their original to their 10 times dilute, and then they had to go from 10 times dilute to twice more dilute. So in this case, they could look at the dilution factor of each step. And so dilution factor, what's going to end up happening is we have two volumes that are being divided by themselves, and we're going to end up with this having no units on it. Oftentimes when things like don't have units, but we say it's like a factor, it's still something that we're going to multiply back. If we find that the dilution factor is 10, we might write it as 10x, just to know that it's like a multiplier thing. And that will help you remember that it needs to be multiplied back into our cell concentration equation, right? So what I have next are is practicing with some of that, like how do we read problems and how do we make sure that things are basically what we want from them. So one second. Trying to find a good layout so that you guys can see the whole question. Maybe I should just, there we go. Haha, I'll just, so of course now it decides to change, right? So this is a problem of cell concentration. We know we're looking at cell concentration. You see here, the human cytometer was loaded with a solution of cells, which has been diluted 100 times. And then what is the final cell concentration in cells per milliliter? So we know we are looking at this cell concentration equation. So I know that I need to find an average number of cells. So I'm just going to shorthand concentration as CONC, right? Average cells times my dilution factor times 10 to the fourth. That 10 to the fourth will be true all the time. So my next step would be counting the cells. This particular human cytometer, if you can tell from your screen, has two different colored cells, probably for some that are alive and some that are dead. I'm just going to get a total cell concentration. So on boundary that I'm going to include. If I'm including that one, I decided I probably should include this one up here too. So I'm going to change this to 8 instead, which means I'll need to include this one. 7. Okay. So I have 7, 7, 8. Apologies for changing that. And 5. So I'm going to add all of those up, which is 14, 22, 27 over 4 is about 7 times my dilution factor, which is right here in my problem, 100x. So times 100 and times 10 to the fourth. And so using my handy dandy calculator, I will find that this is 7 times 10 to the 6 cells per milliliter. I'll make this really simple for you. 1 in chat if you would like another practice of that, or a 2 in chat if you think that I should move on. So 1's in chat if you would like to practice again with this problem on cell concentration, or 2's in chat to move on. If I don't see anything else, I'll just move on so that we can hit some other topics. 1 in chat? Okay, thank you, Morgan. Would you like some time, Morgan, to kind of look at this question and try it yourself? And then we can go from there. And then I'll like do it along to see if you got the same answer. So this problem reads, the hemocytometer here was loaded with a solution of cells after 200 times dilution. What is the final cell concentration in cells per milliliter? Okay. I'm going to turn off my camera for a second. I'm still here in the background, but the plan is next to do maybe some dilution factor practice, or go on to an entirely different topic that you guys are interested in. Good time to drink some water. So again, on this one, there's the filled in cells and the unfilled in cells, and you can just count total number of cells. All right. So first thing I'm going to do is count the cells in my major squares and find an average. So 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4. I'm going to count this one. So 1, 2, 3, 4, 5, 6. And 1, 2, 3, 4, 5. So I've got 10 and 13, 23 over 4 is about 6 cells as my average cell count, times my dilution factor, which what is the dilution factor this time? And then of course I'll still have my times 10 to the 4th. $200. Good job, Morgan. 200. So this one's a little bit trickier because now I have a 2 to multiply in there. So this will end up as 1200 times 10 to the 4th. And so writing in scientific notation, right, we want to have a number that's between 1 and 9.99999, repeating nines. And so I need to move my decimal 3 times, which will give me 1.2 times 10 to the 7th of this 3 plus the 4 I already have up there. Cells per milliliter. All right, do we want some practice with that dilution factor equation, or kind of move on to a different topic within our review? We got about 20 minutes left to cover things. And then, of course, you can always stop by your lab instructor's offices over the next few weeks to talk about things as well. I could also bring my video back. Another topic. All right. So, Morgan, we had looked at, I don't know if you were maybe not here yet, but we had, where did it go? So, we can talk about bacteria. We can talk about engram staining. We can talk about results narratives. We can talk about Logger Pro and genetics stuff, or the cell cycle. Any of those sound interesting to you, Morgan? Or it's somewhere where you would like some additional review? The bacteria and gram staining stuff, we can look at like what our gram stain steps are and antibiotic mode of actions and which one's like effective, and kind of how to use those effective, ineffective, susceptible, and resistant terminology. In the results, we can work on assigning letters to our figures. Gram staining steps. Okay. So, yeah, so our gram stain steps, like what are those steps? And then what is kind of like the purpose of each of those steps? So we know that we have 4 steps. So there's 4 steps to gram stain. Do you know any of those steps, Morgan? And we'll compare what it looks like on a gram positive and a gram negative bacteria. Can't recall. Okay, that's fine. So the first thing that we do is we add our crystal violet. And what that is going to do as a purpose is that is going to stain our cells purple. So what a gram-positive and a gram-negative would look like under those just after the crystal violet step is they would look purple, which I'm just going to shorthand perp for the sake of time. So the next thing that we do is we would grab grams iodine. And so what you need to know about these is really about the purpose of the steps and the order that they go in terms of that can help you remember how the process works. You won't have to do a gram stain during the lab practical. So this grams iodine, its purpose is to create a complex with our crystal violet, which I'm going to shorthand as CV. And this complex, it makes the molecule larger, right? And so we know that gram-positive has this thick peptidoglycan layer that's hard for large molecules to get through and move out and stuff. But because we've already stained with crystal violet, the crystal violet's already soaked in there, and then the iodine also, I'm using my camera as if you can see it. So that the crystal violet will soak through that layer of peptidoglycan, and then the iodine comes in, but it creates this complex, so it's harder for that complex now to go back out. And so these stay purple for both gram-positive and gram-negative. This next step, this is our key step right here. And this is our ethanol rinse. So we're using ethanol to kind of rinse our cells. And so ethanol is an alcohol. And there are similar alcohols that are in hand sanitizer. So I always try to remember after I've used hand sanitizer, my hands always feel so dry, right? And so part of this is it dries out our cells. And now that we've learned in lecture about osmoregulation and hydrotonic or hypotonic and hypoosmotic solution, we know that when cells get dried out, this means they shrink. And so both our thick peptidoglycan layer in the gram-positive and the thin layer in the outer membrane of the gram-negative are going to shrink with the cell. So what do you think that shrinking is going to do with our crystal iodine complex in our gram-positive bacteria? Or what's going to happen with our gram-negative bacteria, where we know that outer membrane is made up of lipids, right? And the reason our hands are normally soft and not dry is because of oils, which are also lipids. It will now create a difference in the colors, right? And so now our gram-positive will stay purple, and our gram-negative, because of also what I was starting to say about the oils, is that outer LPS, that lipopolysaccharide layer on gram-negatives will be stripped away, right? So our hands normally have some natural oils on them. When we use that hand sanitizer, they get dried out, which means our cells shrink, but also it stripped away those oils. So that outer LPS, right, this is a lipid or oil layer you can think of, on gram-negative is stripped away. And so when we shrink, right, I've talked about how this crystal violet complex is a larger molecule. It's going to get trapped inside the shrunken peptidoglycan layer of gram-positive, where everything in gram-negative will be able to be rinsed away and create clear. But we know when we look at gram-negative, it's not clear under the microscope, is it? No, it's got a color, right? And we know that gram-positive ends up purple. And so, and that's mostly because as clear, the you can't really see clear cells under the microscope, so we have to give it a color. And so basically what we do is we use safranin to create this counterstain to make this pink. And so that's how that part works. So the other thing, right? And so now we know that pink is gram-negative. And sometimes people say this is red, and that can kind of help. Like if you remember pink and red are the same things, then you can think of purple being positive, right? This P connection. And then if you think of red instead, you know that this would be negative, or maybe like the E connection, or, you know, maybe this N in pink and the N in negative. Help you remember which color is which for gram-positive and gram-negative. So relatedly to the gram staining, right, is talking about our modes of action and being able to use terminology. And so we will, this did not look like this in PowerPoint, but we use the terms effective and ineffective, or susceptible and resistant. And it's really important that we use those in the right way, right? And so effective and ineffective are used when we're talking about an antibiotic and how it worked against a gram-positive or gram-negative bacteria or a specific species of bacteria, like against M. luteus or against E. coli, right? So you would say something like, and actually this doesn't just have to be antibiotics, this could be anything that you apply to a bacteria. So it could be essential oils, it could be spices. That's what some of you did your independent projects on, right? So it would be along the lines of, you know, tea tree oil was effective against gram-positive bacteria because of it creating that zone of inhibition that we see, right? So we kind of say that effective, ineffective are used when we're talking about the antibiotic. Conversely, right, we use susceptible and resistant to talk about the bacterial species. So when we have that larger zone of inhibition, we could say that gram-positive bacteria are more susceptible to the oregano oil because we saw that larger zone of inhibition that we would get data from. Now, a smaller zone of inhibition would be an antibiotic is ineffective or a bacteria is resistant. So that's kind of how that works. So the two antibiotics that we looked at in lab this semester were penicillin and streptomycin. And so that penicillin, right, it inhibits the peptidoglycan layer formation. So PG is for peptidoglycan layer. And so would you think that that's going to be more effective a gram-positive bacteria or gram-negative bacteria? If we're inhibiting that peptidoglycan layer formation, and that's important to one of those different bacteria types. Whereas streptomycin inhibits protein synthesis, and we know that protein synthesis happens inside the cell, right? And so streptomycin must be able to get inside a cell. And so which of our cell types is easier to get into? So the answer would be that penicillin would be better against a gram-positive. We know that gram-positive have thick peptidoglycan layers. And our gram-positive bacteria this semester was M. luteus. So again, Morgan, I find that a great way to remember which of these works against which type of bacteria is penicillin works against gram-positive. So like all those P's go together. Purple, positive, penicillin. Only hard part is remembering that it's M. luteus, and I just think that the T in M. luteus looks like a positive sign. Right? So if we inhibit the peptidoglycan layer formation, it's going to be more detrimental to a cell that has a large peptidoglycan layer rather than a small peptidoglycan layer. And our gram-positives are what have those large peptidoglycan layers. And streptomycin works better against gram-negative because it can get into the cell more easily from having that thin peptidoglycan layer. And we can remember that E. coli is our gram-negative bacteria sample by looking at our E, and kind of like the 3 lines of the E all create negative signs. And then less so important, but these antibiotics do fall under different classes of beta-lactam and aminoglycosides, but that's not as important for the lab practical. So I think what I'm going to do is kind of wrap up, and I'm going to show you the skills that you're going to learn in lab next week that will potentially be on your lab practical, just to kind of give you a review of what else you'll want to kind of think about. And actually, let's just look at the other skills that I had prepared so you just kind of know what you might want to study. So writing a results section, making sure you have proper narrative structure, being able to assign letters to figures based on that 2-key p-value output, and be able to write a proper figure caption. Genetics, knowing how to identify if something is heterozygous or homozygous on a gel, or with a genotype provided to you, that's the letter designation. And then being able to determine paternity inheritance from a gel. So that would be important things. Related to enzymes in Logger Pro, kind of like remembering when we want to calibrate the spectrometer. And so that's once we have the substrates and our buffer, so the water, the glycol, the hydrogen peroxide into our thing, because glycol has a slight color. We cannot see it with our naked eye, but the spectrometer can sense that, and so we need to set that as our 0 point, right? And then making sure we hit that collect data button will be really important once we've finished the calibration, but before we're putting that enzyme in, so that we get all of the data that we want. As well as, this is good practice of concentration volume. So there's some resources on Canvas for practice. A lot of times you can just look up concentration volume questions, although some of them might get more into molar mass and like chemistry related stuff and not stay too on biology, but you can always ask your lab instructor maybe to come up with some additional practice problems for you as well. So this is highlighting where we find the initial rate of reaction, right? First, we want to highlight the correct portion of our figure, like graph thing that comes back up, and then that is the button that we will be hitting to get that initial rate of reaction. And which of the numbers that comes up, right? There's a bunch of numbers that will come up, is the one that we want to like kind of write down and decide that's the one. Basically, which of the numbers that's going to come up is going to give us our initial rate of reaction. And hopefully you were thinking M for slope, right? We're looking at a rate of something, so we want our slope of the line. I'm going to skip that, but this slide is highlighting an area that you would want to kind of highlight as an initial rate of reaction, right? So you will see that that green line has a steep incline from below 0 to above 0. That's you putting your cuvette back in that spectrometer. And so then you want to be just after that bump is where you would want to get an initial rate of reaction from. And hopefully you won't have lines looking like the red and blue one for your lab practical, and will be nice and give you some slower enzymes to work with. But sometimes the enzymes have a mind of their own as well. The cell cycle, making sure you know, kind of like be able to determine the chromosome number and be able to draw or identify those stages and whether something is haploid or diploid, or if we give you 2n equals 6 in a certain phase, being able to draw that properly. And so as I was talking about, these are the additional skills that we're going to look at in lab next week. And that's using a glucometer to measure blood glucose levels, understanding the blood glucose homeostasis feedback loop, looking at some heart anatomy. You learned a lot about heart function in lecture, so hopefully that will be helpful to both lab next week and your lab practical. But then also understanding endocrine versus exocrine function of the body. So probably, you know, take a screenshot or write down the things from this to kind of, you know, like, okay, these are the things that I'm going to need to know from lab next week for my practical the following week, right? And so as I had said before, right, they're Sundays 5:30 to 8:30 here in Johnson, 244. There are TAs available for you to practice any, you know, micropipetting skills, microscope skills. So they're here, you know, every Sunday until we finish lab. So both in the next 2 Sundays, you can stop in and chat with them. And so I hope this review session has been helpful to you. And as always, right, lab instructors' offices are open for you to keep chatting, and there'll be some other different slides that we put up as well. I will put this slideshow on Canvas. There's another set of review slides we will put on Canvas. So thank you for coming, and good luck on your quiz tomorrow, and I'll see you around. Bye, everyone.